Does anyone have any questions to get started? Nobody has any questions? Right. Yep. Hopefully everything works out well.
Hello, Professor. Are we going to use the Zoom for the lecture exam, or will it be as normal?
If I can get it all figured out in time, I'll send you the information. So hopefully I'll get that set up. Because I was really hoping to be able to start seeing you guys, but it just doesn't work well with Collaborate. So.
But for the lab, we're still using Collaborate, right?
Just so that you can ask questions. You don't have to turn your cameras on.
Oh, I thought last week we said, last class we
did, but this morning it didn't work well, so we're not doing it.
Oh, okay.
If you really want to, you can, but it doesn't work well. It can't handle all the different cameras, so. Well, red, it's more of a fuchsia color, but reddish purple, purplish red.
Are you gonna go over the unknowns today, or?
I'm gonna grade them while you're taking your exams.
Oh, okay, thank you.
SFS negative is blue. SFS positive is, yeah, negative is blue. Positive is kind of fuchsia color. So what do we call the flow of information in a cell? Right, the central dogma of biology, right? Yeah. And so what do we call the process of making RNA from DNA?
Transcription.
Right, transcription. And what do we call making protein from RNA?
Translation.
Yep. And then we learn that we can go backwards at one of these steps. Where can we go backwards and how can we do that?
I think it was you could go from RNA to DNA with reverse transcriptase.
Yep, with reverse transcriptase, right? So an enzyme originally discovered in viruses, and that allows us to make DNA from RNA. What enzyme makes DNA? What enzyme makes DNA?
Polymerase. DNA polymerase.
Yep, DNA polymerase. And what enzyme makes RNA? RNA polymerase, right? And they both have the same problem. They can only synthesize new DNA or RNA in one direction. And what direction is that?
3 to 5.
Well, the new DNA is added 5 to 3.
Oh, 5 to 3.
Right? 5 to 3. But the 3 to 5 prime template is the one that will be used, right? So when you write the complementary DNA sequence, what would be the complementary DNA sequence to this piece of DNA? Well, so, right, CT, AA, TC, T, right? But if this is RNA, what would the sequence be? Right, CU, AA, UC, U, right? So you should be able to write the complementary messenger RNA. And then remember, every group of 3 nucleotides is a codon. And I'm not going to show you the chart, but remember the first nucleotide, you look on the left-hand column, middle nucleotide, you go across the top to find it, and then you triangulate to find the third one, to find the codon. Right, so on the chart, now when DNA gets replicated, of course, you remember that one of the new strands will be synthesized as one long continuous piece, and the other one's done in little short pieces called Okazaki fragments. Remember, that's because the DNA polymerase can only replicate into 5' to 3'. Only one of the template strands is in the correct orientation, and the other one, remember, has to loop around. And there's also a little bit of primer in here, right? Some RNA primer. And remember, that creates a region that's double-stranded, so DNA polymerase can hop on. And eventually the primer is going to be removed. Everything will be sealed and joined together. Right? What do we call the things that actually translate to make protein? What are those little machines called? What are they? What makes proteins? Ribosomes, right. What were the 3 mechanisms of horizontal gene transfer? It's easier to say it than to type it. Horizontal gene transfer.
It had like protein, DNA. I forgot the order.
But remember the 3 mechanisms of horizontal gene transfer, and we did one of them in lab.
Yeah, is it transformation, recombination, and conjugation?
Right, so transformation, conjugation, and transduction. Which one requires the virus in order to happen? Which one requires a virus? Which one?
Transduction.
Yep. Remember, transformation normally happens with little naked pieces of linear DNA, but in lab, we don't use linear DNA, right? What do we use in lab?
Plasmids.
Plasmids. And bacteria don't normally absorb plasmids, so we have to force them to do it. We make the cells competent, and how did we make the cells competent?
By shocking it.
Right, the heat shock, along with calcium chloride, makes the membrane permeable, right? And that allowed the plasmid to get in. Now, conjugation, remember there were two cell types that could conjugate, F plus and HFR, right, cells, and they conjugated with an F minus. Remember, the HFR and the F plus cell can produce a pilus, which grabs the F minus. But remember, when an F plus conjugates with an F minus, we get an F plus cell, right? The F minus becomes F plus. It's fertile, it can conjugate. But when the HFR conjugates with an F minus, the end result is still F minus, right? The recipient cell does not receive a copy of the fertility plasmid, but may have received some bacterial genes that were on the bacterial chromosome. So it may have acquired some new characteristics. Oops. Yeah, that's not. I don't like that. Start again. So what type of DNA is this? Eukaryotic or prokaryotic?
Eukaryotic.
Eukaryotic, right, because it has introns. So you remember the final messenger RNA, all the introns are going to be spliced out, right? So it's all exons. So remember when we talked about cloning eukaryotic genes to prokaryotic cells? And we said we can't take the gene with introns and stick it into a bacterial cell because the bacteria cannot remove the introns. So we said we had to isolate the processed messenger RNA transcript with all the introns removed. And then we use reverse transcriptase to make a DNA copy. And so now we have a clean gene. And so now you can tell me the last step. How do you stick that gene into bacterial cells? So how are you going to stick that clean gene into the bacteria? Nobody knows? You do know. We just talked about it. No, using a pilus, that's too much trouble. That's too tedious. It's a lot easier way to do it. What's the easy way to do it? Transformation, right? We just learned how easy it is to stick, genes into bacteria using transformation. So that's the last step. Isolate the process messenger RNA transcript that has all the introns removed, then use reverse transcriptase to make a DNA copy without introns, and then use transformation to stick that copy into bacterial cells. And hopefully they'll start making, right, the protein that you want. So which cell type can make proteins faster, prokaryotes or eukaryotes? Right, prokaryotes. And do you know why, Bryant? If you don't know, Bryant, just type a question mark if you don't know. Right, so in prokaryotes, transcription and translation take place in the cytoplasm, whereas in eukaryotes, transcriptions in the nucleus and translations in the cytoplasm, so there's a delay as the messenger RNA is transported from the nucleus to the cytoplasm. But in prokaryotes, both transcription and translation can occur simultaneously. And so prokaryotes can make proteins faster. What do we call changes in the DNA? Mutations. And remember, mutations can be beneficial, they can be harmful or neutral, although most mutations tend to be harmful. Every once in a great while, a mutation is actually beneficial, and it's the accumulation of beneficial mutations that allows for evolution to occur because it allows for organisms to adapt and survive. And we talked about point mutations like a missense mutation. Oops. Where one base is changed, one nucleotide is changed, and it changes the codon into a different codon, so it codes for a different amino acid. And then we had nonsense. What's nonsense? And not the stuff you're going to write on your exam, but what's nonsense? What kind of relation is that?
When the codon is supposed to be an amino acid, it turns into a stop codon.
Right. So the codon changes into a stop codon, and that produces a short protein, like Christiana said. So a short protein. And that's most likely not going to be functional. So if it's a critical protein, that cell's going to die. And then we had frame shift mutations, where the base change is either inserted or deleted, and that changes all the codons afterwards. So they're all different amino acids, and that's almost always very, very bad. And mutations do occur naturally. They occur spontaneously, right? The polymerase enzymes are not perfect. But you can increase the rate of mutation by using a mutagen, a chemical or physical agent. And some physical agents we talked about, radiation like gamma rays and X-rays, UV radiation. What kind of damage is caused by UV? What kind of damage is caused by UV? Well, that's in us, right? But the DNA damage are the thymine dimers. Right, so thymine dimers. A few dimers can be repaired, but too many causes too much damage. So then we talked about a classification. And remember all the taxons, all the groupings. The largest one was domain, and then kingdom, phylum, class, order, family, genus, and species. Or damn King Philip came over from Great Spain. And mostly we use the genus and species. And so when you write a name properly, Q, like Thermus aquaticus, you capitalize the genus, species is lowercase, and since I'm writing, I'm supposed to underline. If I were typing, I would use italics, right? Yes? And then we talked about the origin of mitochondria and chloroplasts. And where did they come from? Where do mitochondria and chloroplasts come from?
Wasn't it that they came from bacteria? Like one of them swallowed another one.
Yep. And what do we call that theory?
Endosymbiotic.
Yep. So the endosymbiotic theory, the idea that one cell swallowed another a very long time ago, the cell that got swallowed, the symbiont was not destroyed, but they lived together. And over time, the symbiont specialized for energy production. And that's why we see so many similarities between mitochondria, chloroplasts, and bacteria. So when we classify microorganisms, right, we want to name them, right, so we can help to identify them. And we know that we can perform a variety of techniques to do that. Simple ones like doing stains and then doing biochemical tests. But we often like to, run gels, to separate DNA, but we may not have enough DNA to work with. So we need a way to make more. And what does PCR stand for? What's PCR?
Is it polymerase chain reaction?
Yep, polymerase chain reaction. And there are 4 ingredients needed. Obviously, we need our template DNA that's going to be amplified. We need all the nucleotides, right, to make it. We, of course, need the enzyme DNA polymerase. And we need primers, right, because we have our double-stranded DNA, our template DNA. How do we separate that? Because we have no helicase, so how do we pull the strands apart?
With heat.
With heat, right? So we use heat. and we pull our strands apart. The primers bind, and polymerase makes new DNA, and we've doubled our DNA. And then we heat it up again. Now we have 4 template strands. Again, the primers will bind, and polymerase will replicate to make more DNA, and we've doubled it again. And we repeat this over and over and over again, 30, 40, 50 cycles, right, to dramatically increase the amount of DNA we have. But there is a problem, right? 95 degrees Celsius, that's almost boiling. And we're using an enzyme. What's the problem with the enzyme?
Can get denatured.
Right, denatured. So you really don't want to add a DNA polymerase 30, 40, 50 times because, one, that's extremely expensive, and it's extraordinarily tedious to have to do that for each cycle. So how can we make this process efficient and economical?
Using thermophiles.
Well, we got our DNA polymerase from a thermophile, right? And so that means that the polymerase is very thermostable. It's very heat stable and does not denature because it came from a thermophilic organism. And that organism is one that I just showed before, Thermus aquaticus. Let's see. And the brand name is Taq polymerase. But the whole reason why by PCR is efficient and economical is because you add that polymerase once, and it's good cycle after cycle after cycle because it does not denature. So any questions on PCR? We went over a bunch of different kinds of bacteria. Remember I said the table where all the organisms are underlined. You know, those were the ones that were most important to focus on. Not the only thing, but the most important. And then we had the enterics. What are the enterics? Remember the enterics? Right, gram-negative rods, mostly. Remember facultative anaerobes. And then we also talked about staph and strep. Most of the rest were just examples of the different things that we talked about. Just make sure you focus on the table with the underlined organisms and characteristics, as well as the enterics and staph and strep. Then we talked about antimicrobial drugs, right? Antibiotics, which technically are chemical substances produced by one microorganism to inhibit or kill another microorganism. Although we have a semi-synthetic and synthetic antibiotics that have been created. And we talked about targets. So there were many targets on the bacteria, or that the bacteria have. And what are some of those targets? What are some targets they have? Right, the cell wall, cell membranes, DNA replication, transcription, translation, a critical metabolic pathway, which if you can find one that's unique to the bacteria, remember we can use a competitive inhibitor. Right, to block that critical pathway. And in particular, the prokaryotic ribosome is a kind of a favorite target because we have many different antibiotics which target ribosome, because you remember the prokaryotic ribosome is different from the eukaryotic ribosome, so we can exploit those differences. And we have many kinds of antibiotics like tetracycline, right, which attack protein synthesis or translation. But when it comes to the most selective target, what is it that the bacteria have that our cells do not? What do bacteria have that our cells do not? Right, the cell wall. So an antibiotic that targets the cell wall would be the most selective, right? Because it would leave our cells alone completely. And we have many antibiotics like the penicillins. Remember the penicillins? Right, which target peptidoglycan synthesis. Now penicillins, what group of bacteria do they target? What specific group?
Is it the gram-positive bacteria?
Right, so gram-positive. So since it only targets one group, what do you call that kind of antibiotic? Only targets one group.
Narrow spectrum?
Right, the narrow spectrum. But something like tetracycline, it targets gram-positives, gram-negatives, rickettsias, and chlamydias. So what would you call that? Broad spectrum, right? So a broad spectrum. Now we know that some bacteria can resist the penicillins. What enzyme do they produce to resist the penicillins? It's the same enzyme that we use to determine transformation, right, to select for the transformed bacteria. Right, so beta-lactamase. And so if the bacteria have beta-lactamase, they're not only resistant to penicillin, they're resistant to amoxicillin, oxacillin, ampicillin, even some of the cephalosporins. And so we call that cross-resistance, right? When bacteria are resistant to more than one antibiotic. And that's good for them, but bad for us. We also talked about efflux pumps as a common mechanism of resistance because they pump out all different kinds of chemicals, right? Antibiotics, disinfectants, antiseptics, so they can work against a wide variety of things. And it's the misuse, abuse, and overuse of antibiotics by humans which are selecting for these highly resistant bacteria. We talked about antifungal drugs. What was the problem with antifungal drugs and finding a good antifungal drug? What was the problem? Right, fungi are eukaryotes, and we are eukaryotes. So at the cellular and molecular level, we're very similar, right? So trying to find differences to exploit was not easy. And they did find a membrane component, ergosterol, that the fungi need that we don't. And when it comes to antiviral drugs, most of those are synthetic drugs. And the idea behind most antiviral drugs is to either stop the virus from replicating or block the virus from infecting to begin with. But they're not really cures, at least in most cases. The antiviral drugs are not cures. We talked about methods of control. We talked about some physical methods as well as some chemical methods, the antiseptics and disinfectants. For the physical methods, we talked about moist heat, right, versus dry heat. We said that moist heat of the water helps, so you need lower temperatures, like 121 degrees Celsius, for less time as compared to dry heat. You need much higher temperatures, like 170 degrees Celsius, for a much, much longer time. We said that we could use radiation, right? Ionizing radiation is good because it penetrates deeply into something, so you can sterilize the whole thing. UV, we said, was only good for surface sterilization because it does not penetrate deeply. If you have a product that cannot be heated, like an antibiotic solution, you can filter sterilize it. So, you have to consider how you want to sterilize your product in such a way that you won't damage that product. Maybe you gas it, right, with ethylene oxide gas. When it comes to antiseptics and disinfectants, on what do we use disinfectants? On what do we use disinfectants? What do you use Clorox bleach on? Or Lysol disinfectant spray? Right, we use it on inanimate objects like surfaces, tabletops, floors, instruments, things like that. And antiseptics we use on living tissue. Now, as we're going to learn later on in lab, when we do disc diffusion, sometimes you can use antiseptics and disinfectants interchangeably, like alcohol, but most of the time you cannot, right? Most disinfectants like bleach, Lysol, you don't use on living tissue because they're too harsh. And some of our antiseptics are not really strong enough or last long enough to really work well on surfaces. So most of the time you cannot use them interchangeably, but sometimes you can. And when it comes to alcohol, remember that was our example where full strength actually does not work. You need to have some water mixed in with the alcohol, and that allows the alcohol to penetrate the cell so that it can denature proteins and dissolve those lipids. If your alcohol is above 95%, it just causes the cell wall to immediately denature and form a protective shell. And so you can't kill that way. We can use heavy metals like silver, mercury, and copper, but those are only applied topically, right? You don't take them internally, you don't, swallow them or inject them or anything like that because they're too toxic that way. And when it comes to killing things, you have to take into account characteristics the microorganisms have. If they produce spores, you have to make sure and use something that actually kills spores. Maybe they have an unusual cell wall that's thick and waxy like mycobacterium. So you have to consider what kinds of microorganisms you're trying to kill and also the environmental conditions, right? Because not all the antimicrobial treatments work equally well at high temperatures or low temperatures or high or low pH. So you have to consider a variety of factors when you're trying to kill microorganisms. That's basically it for a general review. Do you guys have any specific questions about lecture material? No?